Thank you so much. It's a real honor to be here. This has been a fantastic conference, and I really appreciate the opportunity to at least help someone with their bingo card with an NIH 1000 pangenome check mark on it as well. As mentioned, I'm going to talk a lot about reference genomes. This is something that's pretty foundational to almost everyone's project and probably almost every breakout session we're going to have at this conference. It was foundational for changing the way we do human genetics and genomics, but I want you to take a moment and no longer think of it as maybe a biochemical structure, as I'm showing you the double helix, but rather something that's more akin to a coordinate-based system. You see, the human reference genome, whether you're using HG38 or HG19, is actually a coordinate-based system that can be downloaded freely and openly wherever you are on the globe. And for the past 2 decades, there have been billions of dollars and international support to build an incredible amount of annotation and information on top of this reference genome. This is a foundational tool, not just because of the sequence, but because all of that information that sits on top. And that has guided our understanding of basic science, clinical genetics and genomics, as well as our own human histories. However, this tool is over 2 decades old. I'm pretty sure many of us don't even have a computer or a phone or medical equipment that we've been keeping for that long, and it's very much due for an update. The argument that I'm going to make that I hope you take away from my talk is that it's time to modernize the human reference genome. This is an image shown from the celebrated International Human Genome Project back in 2003, and I'm hoping what your eyes catch is, of course, these bright red blocks that I'm showing to you. These are huge gaps that represent over 200 million bases that were known to exist when we had celebrated the first complete, in air quotes, human reference genome. Of course, we celebrated in 2022, really through the leadership of the Telomere to Telomere Consortium, which I co-led with Adam Philippi, the first release of a complete human genome. Now those red blocks that I introduced before, you can see on the cover here, actually celebrating new sequence that had been introduced. So now we can begin to study chromosome biology end to end, an entire chromosome, every base in between. This gives us new resolution into fundamentally new biology like segmental duplications, and that often includes duplicated gene families, subtelomeric regions, entire acrocentric short arms that had been missing from our studies, as well as the centromeres themselves. Now, this was performed mainly through a lot of innovative new technologies and also through the sequencing technologies of long read development. And so this was a combination of PacBio, HiFi that came out at the time, as well as Oxford Nanopore Ultralong. With these more complete T2T assemblies, we just instantly started seeing better results in terms of determining variants. New discoveries were shown. When you map 1,000 genomes data back to a more complete assembly, you're going to find more novel variants on the order of hundreds of thousands. There are errors, persistent errors, that were found in HG38. And when we have a more accurate and complete reference, we found that we eliminated those errors, and we also saw that there was a 12-fold reduction of false positive calls in medically relevant genes. Now, before you tell me these newly introduced regions are highly repetitive and are not useful for short-read data sets, let me emphasize that we're even seeing gains of new knowledge when you use ancient DNA or fossil DNA's mapping to this reference. However, one genome, no matter how perfect it is, cannot possibly serve as a reference for this community. And that's because it is insufficient to represent the genetic diversity that we know we have across humanity. And this is important not only for understanding the variation and ensuring that we avoid mapping bias and genetic bias in our work, but also because a lot of fundamentally important medical regions like HLA, SMA, and CYP26 is actually really important for our understanding of medical disease. Also, we've been spending a lot of time as a community on small stuff, single nucleotides, small indels, and I want to make sure you leave this talk understanding that there's a lot of information on structural variants, or these are the larger regions that we know are highly variable in the human population. There are tens of megabases of these common polymorphic sequences missing from any one individual reference, And no single reference assembly, even a complete one like a celebrated T to T one, is optimal for all people. That introduces reference biases. If you were to take short-read data sets where you have a benchmark set, you know the structural variants, and you remap it to HG38, 70% of those events are going to be missed by the standard methods of calling variants. So if we think about HG38, or I hesitate to even mention HG19 that I know many people are still using, we can think about it as being kind of a composite, where I'm showing it's largely represented by one individual's donated genome, but we have 19 other individuals who donated as well. But I'm trying to bring your eyes to see it's a single reference map, a linear reference map that shows that type of composite information. When we talk about a pangenome, what we're talking about now is going through and trying to represent humanity by thoughtfully selecting reference genomes to try to represent common variation that we see across the globe. And by doing that, we're able to actually generate what we call a tube map here, or maybe a metro map, where you can begin to see the areas of your genome that may share a haplotype with someone else, and we can begin to identify variants, single nucleotide variants, inversions, insertions, deletions. This can all be modeled inherently in this new data structure. Today, I'm here on behalf of a large consortium. This is called the Human Pangenome Reference Consortium, or the HPRC. This consortium was a call to action from NIH. Our goals are really to build this core resource, and in doing so, we need to make high-quality assemblies, T-to-T assemblies, the ones that are complete and highly accurate for at least 350 diverse humans. That would be 700 haplotypes if you consider both the maternal and paternal haplotypes. In doing so, we create a map of variants, a map of these haplotypes, and a new reference data structure and coordinate system that we're hoping will be transformative for the way that we do genetics and genomics in the future. In order for this to be successful, that means we have to nucleate and foster an ecosystem of new tools. If you are a new early career scientist, this is an incredibly exciting space to be in right now. In order to promote adoption and guide deployment, this is all on our list to do over the next 5 years. And we need to establish a model for long-term growth, and in order to do this, we need to be a global product that benefits and is also contributed to the global community, and that's something that we are very much invested with over the next phase. So hopefully by this time I've brought a real conversation of why this resource is so important, but before I move on, I really want to emphasize why it is. That's because it reduces biases. That's incredibly important because we know that this type of local sequence divergence between personal genomes and reference genomes can often confound variant detection. It improves calls. It's really important for equitable genome analysis because it allows us to maximize the representation of common variation as well as representation of genomes across humanity themselves. And this new reference, or a global pangenome, aims to cover most of the common haplotypes that can better represent variation that's found in any human sample. So this is an exciting invitation to give a talk, because just 2 weeks ago, our consortium celebrated a big release, and I'm delighted to let everyone know about it here, and I'm so happy to see so many phones going up. So yes, please, I have the QR code here because we have our data download. And I'm going to use the remaining time because I really want to tell you about this release. Here, we're celebrating 470 assembled haplotypes. What I told you before about these more complete T-to-T assemblies, we have much higher continuity and a large number of these T-to-T chromosomes that are being issued. We have gone through new measures and new workflows to ensure improved base accuracy. And we've included long-read RNA-seq. This is the PacBio Connects data for every assembly that is out. This is also a moment where we can step forward and start to also applaud and bring a spotlight to our collaborators in this release. Here, it's wonderful to be in Milan with Human Technopole being one of our key contributors, as well as the University of Tokyo. So as I mentioned before, there's been a lot of enthusiasm about using long reads to try to build better and more complete references. And so this is a moment where I get to highlight that our team is using a multi-platform approach where we utilize 2 smart cells of PacBio reaching relatively about 60x coverage of hi-fi data sets. We use 2.5 flow cells of ONT to reach the 30x 100 kb plus. And we also work with Illumina data sets to reach at least 60x coverage of OmniC. This has become kind of our production data set, and we do this across many production sites in the United States. This is a new world where we're able to utilize software like HiFiism, ONT, to actually start to generate T2T automated assemblies. So what you're seeing along the bottom of the x-axis is each human chromosome, and I'm showing you the counts, or the number of T2T assemblies. When I first started giving talks on reference genomes, I used to give numbers like N50s or the continuity scores, and now the bar is really how many complete chromosomes can you issue at once. It's really astounding. The red is showing you where we're able to take information from that graph, the assembly graph, and make some small but highly confident understanding about scaffolding. Sometimes we even call it grapholding. In this case, so we are able to recover T2T chromosomes with a very small or minor gap. And so what I'm trying to show you here is we have a tremendous number. I do have arrows, though, to bring your attention to the ones that we're not yet successful on. Those are the ones that are acrocentric short arms. They're still with the rDNA locus, where our team is continuing to innovate to try to complete to T2T. I'm also highlighting that we have used trio phasing, we used Hi-C phasing, and we're seeing that no longer are we held by the technology of trio versus Hi-C. We're seeing really lovely results using both. Now, HiFiism ONT is not the only assembler in town. We are very lucky to have a lot of innovators on our assembly team. I would like to also use a slide to highlight Adam Phillippe's team with Virco. We also have issued in our release both HiFiism assemblies and the Virco assemblies that we're using to see how they support each other in terms of their organization and sequence completeness. And I'm also wanting to highlight that Virco has a special module where you can take our Hi-C data. So this is the same data from data release 2, but just run through a separate assembler, which actually has more attentive to how you can bring in these acrocentric chromosomes. And we are seeing now for the first time using Virco 2 that some of these genomes are coming out completely T2T, which means all chromosomes are coming out with a scaffolded chromosome at least. Our consortium's chasing perfection. It's because we imagine people in this audience may be mapping reads to the genome or to the pangenome. And if there's a persistent error there, that could in some ways complicate your analysis and have downstream effects. So we've teamed up with Google Health. This is a project that came out of UC Santa Cruz to develop Deep Polisher. Here, what I'm showing you on the top is a reference assembly in yellow. We've developed software called Pharaoh, which uses the ONT ultra-long reads in combination with the Hi-Fi reads to ensure the placement, the accurate placement of these Hi-Fi data sets across the reference assembly. And then we go through, as I'm showing you with this partition, to go through and identify 100 base pair partitions across the genome. Each one of these partitions then gives us a lot of information to build a tensor. This is base, mismatch, quality score, mapping statistics. And this allows us to have an encoder-only transformer, and it learns from the sequence context. It can track these relationships, and it turns the information that it's learned into a VCF file and allows us to polish every single one of the assemblies. So when you download the data, hopefully later on, if not today, you'll see that these are all polished assemblies, and they really look a lot better than our last release that we performed. In release 1 is in red, so you can see they're kind of a little bit lower in scale versus our release 2, where now we're seeing 1 error every about 500,000 bases. If we look only within the mappable or the high confident parts of the genome, which is roughly 80% of the genome, we see 54% of error reduction. So this is a lot of corrected bases. And when we go genome-wide, this includes a lot of the newly introduced repeats, we see a 7% error reduction. This has been spectacular because we now are able to go in and make not only base level accuracy and improve there, but try to make the best gene predictions that we can. And so to do that, we were using long-read RNA-seq or PacBioKinetics, where now we can start to see not only the full-length transcripts, but also the isoforms as well. And so we did this for all of the different data sets that are issued. You can download the Kinetics data as well. And in this case, we've now integrated it into some of our automated tooling where we can begin to do automated gene annotation, validation, and work with our colleagues in the UK to do careful curation on the more complicated genes. So perhaps the only reason I'm standing on this stage is because I love satellite DNA. That's what brought me into the world of T2T. And whenever I work with my colleagues in the HPRC, I'm always asking, but what about the most challenging, the most repetitive parts? How are they performing? And I have to say, I'm pretty shocked at the quality that we're looking at now. What I'm highlighting here is chromosome 1. This has a massive classical satellite domain. It's about 13.2 megabases in the T2T genome. And we're able to go through now and characterize these different and diverse satellite classes accurately. The reason why I feel confident in saying things like accurately is because we've developed tools like Flagger, which also uses a hidden Markov model, where we're able to take reads and map them to these particular arrays and look for inconsistencies. Look to see if there's some coverage that would say that's out of copy or has too many copies, it's got a reversion, things of that nature. And when we look at release 1, there were a lot of errors in our satellite arrays. And now we're seeing that the satellite arrays are presenting very few errors. And if I showed you the genome-wide, it kind of mirrors what we're seeing even genome-wide. So this is opening a whole new world of trying to understand new biology across millions of bases that weren't previously available to us. Now, these regions that have been missing for a reason, and that's because they're highly prevalent in these tandem repeat classes. So you have a tandem repeat or a satellite DNA that goes head to tail, head to tail, head to tail for millions of bases. And because they're so large and so repetitive and even have structural variations between them, they have been a complete roadblock for our team to try to generate meaningful sequence alignments. So even though we could assemble them, and even though they looked like they were extraordinarily high quality, we couldn't include them in the pangenome. And I'm trying to highlight this with our last work that was published, only using 47 genomes for our release 1. And I'm trying to bring your attention to chromosome 12, where the centromere is a complete gap. This brings me back to the start of the T2T consortium, where we had to stare at these red blocks where there were complete gaps. And I think it's important to highlight that our team's been working to eliminate those gaps once again. So this is once Central Line. This is work developed by Jordan Eisinga from the University of California, Santa Cruz. And here you're working through a multiple sequence alignment method that's on partial order pairwise alignment. This is building from Uniliner, crediting Pavel Pesner's group at UC San Diego. And here the sequences are aligned incrementally, so you can kind of see that they have this tree-like structure. And by doing so, they're able to handle complex sequence variations and rearrangements. We now have these central alignments for all of these centromeres in the HPRC data set, but I just wanted to give you some feel for at least one. Building from chromosome 12, where I highlighted it was a gap before, we are able to make these really meaningful alignments across the centromere now. So as you can imagine, it's a very exciting time to not only deeply characterize and make sure that we're doing these alignments in a way that makes a reasonable and useful reference product, but also how do we start to call variants and report them in these regions that are often left off the map. So release 2, everything I've shown you, was data that was derived from cell lines from the 1,000 Genomes Project. Of course, you're very well versed, I'm sure already, but the 1,000 Genomes Project represents over 3,000 participants that contributed to this project from recruitment sites, 26 worldwide. This is broadly consented. The data is open and accessible for research and for public use. And there are LCLs, or lymphoblastoid cell lines, that are available to anyone around the world from the Coriell Biorepository. And this shared consent of open data and the availability of cell lines is what was allowing our teams to so openly work with groups around the world. These were cell lines that we had shared, once again, through thoughtful consideration with the Human Technopole, as well as the University of Tokyo. If you wanted to see our roadmap, we've already celebrated the first draft that came out in May 2023 of a pangenome. What I'm talking about today is our beta release. This was in May of 2025. And the serious announcement, the one that's the stable release, where I'm going to try to convince you to not use HC38 anymore and to start using the pangenome, is right around the corner, and that's in 2026. And so right now, that same opportunity to collaborate around 1,000 genome cell lines, where we have the shared open consent and the cell lines can be ordered and assembled, this is something that we're now using as a model, because it was so wonderful working with our colleagues at the Human Technopole and the University of Tokyo to expand out now to many different researchers around the world. And so we're collecting a lot of these international partnerships. If you're interested in gaining more information, I also put the QR code here. Please don't hesitate to reach out. These discussions are happening now. Over the next few years, after 2026, as you can imagine, we definitely see it's insufficient to stick with 1,000 genomes, and we are absolutely wanting to recruit outside of 1,000 genomes. I'm also excited to announce the HPRC is actively going through prospective recruitment right now in New York City. This is in partnership with the BioMe Biobank, of which Emer Kenny, who is here, is the PI of this particular project. In this case, the Mount Sinai BioMe Biobank has over 70,000 participants from 160 countries, and we have sequencing data for over 40,000 of those particular participants that do our type of priority-based recalling and reconsent for the work. Additionally, we understand that there are many other pangenomic projects that are going on in the community. And how do we begin to work together, even if that consent is being drafted right now or perhaps is in the process of being drafted? And so as a GA4GH driver project, the Human Pangenome Project, of which the HPRC is a member of, are working together to identify core consent elements for pangenomic studies. In this case, I don't know how many of you are familiar with the GA4GH RUSE team and their efforts to do consent toolkits. Here, we're trying to identify if there are core consent elements that we can identify to support pangenomic collaborative projects, and that way we can advance some of this global reach. Heather Lawson, who unfortunately couldn't be here today, has an e-poster that's going to be describing some of these discussions, and I encourage you all to look at it. Also, this provides an opportunity to talk through other regulatory and ethics work stream like RUSE and future integration of these projects in Europe. Also, I wanted to bring your attention to another discussion that's happening with a lot of our HPRC LC scholars, talking about responsible access to genomic data and who should keep it, the oversight. So far, I've given you a lot of data, but I have to tell you what we're working on now, and that's pulling it all together to build this pangenome reference. I always like it when my colleagues tell me what is a pangenome reference, where it's the assemblies we just went through, the alignment of those assemblies, and all that rich annotation that we're working on now. But there's more than that. Right now, there's a lot of exciting projects that are happening that are utilizing these resources to drive new genome biology. We're seeing questions that are going into DNA modification, because all these long reads have CpG method modifications that can be mined, the transcription information, even the chromatin conformation. And we're working right now to try to improve our workflows for improved variant calling. I think this is a pretty exciting space, and we're showing that pangenomes power the best short-read variant calling methods, either by SV typing or by looking at the structural nucleotides themselves. So with that, I'll go ahead and go through my summary. I hope you're leaving my talk understanding that the human pangenome will help to ensure broader and more equitable representation in the reference genome used across, hopefully, research and medical fields. That this release, too, will offer over 6,000 T-to-T chromosomes, and it adds 1.4 trillion bases of haplotype resolved assemblies. In this process, our team is absolutely chasing perfection, and we're looking for extremely high accuracy genome-wide, with improvements even over the most difficult repeat-rich regions, including segmental duplications and centromeres. There are active efforts for what I'd say T-to-T pangenome alignments, where we don't leave any part of the genome behind. This is an active space. We're under a lot of research and development here, so it's not complete yet, but we're looking at the first positive signs, being able to include satellite arrays like Centraline. So up next, I think a lot of us are thinking about tooling. How can we start to pull this in to make it interesting for clinical and research community members? And by doing so, that means we need to create new applications to improve annotation in our variant calling workflows. So there's a lot of people to thank. Along the tops, I'm listing a lot of our leadership team and members who contribute directly to the project, as well as the faces of the people. I want to acknowledge always our company partners. The way that this consortium succeeds is by being at the cutting edge of a lot of sequencing technology and new innovation, and we're so lucky to have these company partners. And we're also deeply grateful to the thousands of research participants who make this work possible. Thank you so much for your attention. I'll be happy to take any questions.
Thanks so much, Karen, for the great service you do to the broader community with all these efforts, but also for this stellar lecture. This one is open for questions. Please identify yourself and ask your question. So we have a question over there.
Kaur Alasö, University of Tartu. How far are we from using pangenome reference to reanalyze very large short-read transcriptomic data sets such as GTEx, where we don't have samples available to resequence anymore, but we have lots of short reads. We want to do splicing.
Right. This is an incredibly important question, so I'm very glad you asked it. The truth is, when we have our stable release, that is the best time to map huge amounts of data, but just simply because the cost of what it takes to go through the workflows. However, this is a very special moment because, as you can see, we're ramping up. And so what we're working with now with many groups that have these types of large cohort data sets, or how can we start to do a pilot project? Take a smaller subset, use it on this intermediate release, ensure the benefit, make sure the workflows are working, optimize the cost, because right around the corner we're going to have the stable release, then you'll be in position to go. And so this is a very appropriate time to start planning, start doing those pilots, and start getting ready to mobilize now.
Thank you.
Thank you.
One question over here.
Here, Francisco de la Vega, Galatea Valle. This is an amazing work, but has been moving really fast, the technology as well. I thought that initially we had this idea that Pangenomes reference, we will align short reads to covariants, but just a few days ago, I saw a new version of HiAFSM that can now assemble a T2T with 2 flow cells from Oxford, maybe one later on. In a couple of years, we're going to be doing T2Ts for everybody. So how we're going to use the pangenome in that new framework?
Yeah, that's another great question. You're preaching to choir here. I remember back in 2020 when we had the first T2T X chromosome, and even in 2022, it was like, this is amazing. The speed of which technology is moving right now is just incredible. And I think that brings me back to my first slide of why use HG38. We use HG38 because HG38 serves as a fundamental hub of where all that very valuable information is located. And so if we have something that's an open reference data structure, which isn't just sequence, it's not just T2T genomes, right? In this particular case, it's enriched with information because the coordinate-based system where now if there's clinical variants, if there's information, it's kind of embedded within that structure. You could imagine that in the future, perhaps you have your own, T2T initiative where you've now sequenced 1,000 individuals who are from a certain biobank. You could imagine taking this reference sequence, putting it in within your data structure, for example, and utilizing all that rich information. So once again, helping to provide that kind of transmission of information. So in that way, it's kind of more of a sink of reference data sets than the number of T2T genomes. I do envision that the T2T genomes will expand, as you're predicting as well, if I'm allowed to make a self prediction too. I think that that's a very interesting space, and we do have tooling to support the pangenome being massive. Because at this point, we do more personalized pangenomes, so if you're mapping to it, we find the haplotype that's closest to your particular short-read data set. So it allows for that type of larger pangenomic data structure to emerge, where before we were trying to understand how to do that. And I think there'll be even more innovation in that space too, to allow it. Thank you.
I'm afraid we're over time a little bit.
Oh.
But Karen is around, so I encourage everyone who has additional questions to see her right after the session, and I think these are very important discussions. So thanks again, Karen, for being here and your amazing work.